Hello, thank you for attending the presentation. My research has been on assessing the habitats and organism communities of saltwater impoundments within Little Edisto, South Carolina. And in particular, I'm looking at them in the context of potential oyster restoration in the future. So as a lot of the previous presentations have outlined, there are many different salt impoundments, including many saltwater impoundments throughout South Carolina that have been created for a wide variety of purposes, ranging from mosquito control to waterfowl hunting to aquaculture. And one of the questions that I'm hoping to address is it possible to create thriving ecosystems that provide similar ecosystem services to that which you would find in salt marshes or natural habitats, while still maintaining the impoundment structure or taking them out? So one thing that has not been addressed in a lot of the previous presentations is that impoundments have a great degree of variability. Some are more lake-like. They have wide open areas with tons of open water, and then plants around the sides, or in Keegan's case, a lot of banana water lily in the center. But some of them are also more almost channel or stream-like. And so there's a wide variety of conditions that are found in both. So the study site that I'm working at is on Little Edisto Island, and it's a set of 6 different saltwater impoundments that are located in this general area outlined in yellow up there on Edisto Island. And our stakeholders of these actual impoundments, the landowners, have the intention of trying to restore them to an ecosystem that's kind of functionally equivalent to a salt marsh, even if they're never going to quite be exactly what a salt marsh is. And so we want to facilitate with that the introduction of structures to facilitate oyster colonization. Why we want to do this is because the oysters provide both a habitat, kind of hard structure habitat that is not otherwise present in the environment, and they can stabilize the sediment and kind of prevent additional erosion from going into the impoundments or anywhere or getting out of them. And they can also provide a great deal of water filtration that people have observed in both lab experiments and in the field. So in order to kind of figure out what role oysters might play in these environments, and also to figure out what locations oysters would be the most effective in terms of like trying to introduce them, we wanted to look at this from a very holistic perspective. So we're looking at all of these different organism communities, ranging from fish to plants to invertebrates to birds. I don't have them on here, but the idea is that all of these things are potential food sources for birds. And we want to kind of figure out how are these impoundments structured in terms of these food webs, and how would oysters fit into those kind of extensive food webs? So primary questions are, what conditions and organism communities are associated with oysters within these impoundments? How do the impoundment biota and their habitat conditions differ from functional, like natural salt marsh? And also, how do oysters support at this site restoration goals and the ecosystem services that our stakeholders want to facilitate? So the current distribution of oysters at our research site is rather limited because there's not a lot of hard structure for them to kind of settle on if they're colonized. So unfortunately, there's not a lot of oyster data that I'm really going to be able to show you here, just because of how limited the oyster populations are. They just do not line up with many of our randomized plots that we have set up. But this is the general locations where you find it. So for data collection methods, we're doing a lot of different community surveys. For our plant community surveys, we're doing 10 quadrats in 2 transects that are approximately 1/5 of the impoundment length apart from one another, about equidistant. And then measuring percent cover of plants within the site, and so on. We're taking water quality parameters such as in situ data measurements using EXO2 data sans, so salinity, DO, all of that, as well as processing water samples that we collect from the site for bacterial loading, nitrate, phosphate, ammonia, so on and so forth. So for the plant community surveys, one thing that you'd probably go out here and be able to say, obviously, but our data shows that you do have these kind of two distinct community types. What people normally would consider like a low marsh environment, where you have primarily Sporobulus alterniflorus, which is salt marsh cordgrass. And then these areas have extensive mudflatty sort of locations where they have higher tidal inundation. And these are probably sites where oyster introduction would be reasonably possible. Whereas we have these more diverse, kind of slightly more upland high marsh community sites where we have a lot more plant species in the community and a lot less tidal inundation. So we tend to see, this would not, of course, be a location where oyster introduction would be suitable. So for our plant community questions, we're also looking at seasonality as well and trying to see, do plant communities at these impoundments and at the reference sites change throughout the seasons? In addition to, are impoundments versus reference salt marsh sites different from one another? Are these, are the plant community structures similar? Now, in terms of our models that we created, we see that season is not really a huge factor in the total cover percent of plants at any of the sites. Impoundment is a massive factor where a lot of the more open impoundments that have a lot more inundation with water are very, they have very low plant cover relative to sites that have much less cover. Restoration versus reference is not a strong predictor of plant community assemblage or of the total cover. They're very similar. We do see in this ordination plot that Sporobulus alterniflorus is kind of on the right of this NMDS plot. We have elevation at the center here as our predictive variable. So you can kind of see that basically what this tells you is that Sporobulus alterniflorus is generally found at a little bit lower elevation than the other plants that are there just because it has a much higher tolerance to being inundated with salt water. So for our bird surveys, we follow along this kind of distinct transect, which is more like a bunch of smaller transects. Surveys being conducted between sunrise and 10 a.m. Just to be consistent, we know that bird activity is going to vary throughout the day, so we wanted to be consistent with when we go. And we go to the Russell Creek mitigation site, which is our 6 impoundments, and also the reference salt marsh sites. We have a line transect that is generally here is about 850 meters. And then every time we record a bird, we record our position, the location of the bird, and then the direction and distance to that bird so that we can record where they're present. So our questions here are similar to what I had for the plants, where what birds are using these impoundments? When are they using them? Is there like a seasonal shift in abundance or species occupancy? And looking at the impoundment versus reference salt marsh sites and saying, are these bird communities different from one another? And this is basically a graph, like an XY plot, showing the distribution of where birds are located at these sites, basically where we've observed them. And the primary thing to note here is right along these berms, because these are forested / vegetated berms that have a lot of songbird activity on them because they have higher vegetation than the surrounding salt marsh, which is often rather absent of most of the songbirds. And then the waders cluster along with the waterfowl in these open water areas or locations where there's kind of these little pockets of mudflat where they're able to go out on. So Oxyte's provided some wonderful drone photos that I'm going to look at here. So we have shorebirds and waders that appear primarily in these open areas, songbirds that are generally clustered around the berms and then around these kind of adjacent locations, as well as some areas in the center where you have a large amount of dead trees and other debris that they tend to roost on. And then marsh use is kind of limited in terms of what species are actively going out into the Sporobulus and the other species actively using them. So we see marsh wrens and red-winged blackbirds there most frequently. One of these images just did not show up for some reason. Impoundment 2, that is, if I go back, let's see, way back. This impoundment here, it tends to be dry most of the time, and we've found a relatively low amount of bird occupancy at that location, which we think may be the result of that. There's really not a lot for many of those types of birds. And then this kind of isolated pond site has more divers than we would see at the other locations. It's especially at low tide, it's deeper, but generally it's a very closer to like a normal pond type of environment. And we see that there's a wide range of, in terms of median bird abundance. I'm still in the process of calculating the actual like population densities of birds at these locations. I'm just using median bird abundance from our surveys for now. But the main point is, especially in songbirds, there's a huge degree of variance because sometimes we'll see individual birds, but sometimes we'll also see flocks of like 50 birds in a location, and that results in this wide distribution here. Main point to get across is that this impoundment 5, the largest impoundment there, that has the most mudflat habitat, and also during the spring season is when we tend to see the higher abundances of shorebirds. Waterfowl are almost absent at our sites. We see them very rarely. Pretty much pond is like, that's waterfowl. So that's like an observation of 1 or 2 birds. And then the waders tend to congregate around impoundment 5 and the areas where they have access to habitat they want. So we see there's not a strong abundance factor between sites. There's not a strong abundance factor between seasons, and there's no strong interaction between them. So we see similar numbers of birds generally between the different impoundments, but the season and the individual impoundment do affect what species we see at those locations. So for our fish and invertebrate surveys, we're kind of following a bit of a similar method. We have active sampling with seine netting and cast netting, which we take along randomized transects similar to the plant survey, and invertebrate samples where we use sediment core samples similar to what Tyler and Drew, or Ty and Drew did for Keegan that you got to show there, same method, and we use crab traps as well for the invertebrates. Research questions are the same. How are they similar between impoundments? How are they different based on season? And are impoundments and reference sites different from one another in terms of assembly? So here, the primary findings, if you don't take anything else away from this, Fundulus heteroclitus and Cyprinodon variegatus, which are mud minnows and the sheepshead minnows, are the most abundant fish species across all of the impoundments, pretty much without exception. And a few species are found at higher abundances at the pond site, which has a lot more vegetation cover. And then Gobius homobos and Gormitator maculatus, as well as a couple other species that are on their most recent survey that I haven't put on here yet, only occur at sites with oysters and other shelled organisms, along with habitat conditions that support them. And I've seen them only at reference sites in impoundment 5. So in terms of our model outputs, we see that there is a huge effect of abundance, a huge effect of impoundment that we sample on abundance. So there's a wide range of abundances of fish that we catch between the different impoundments. There's a strong effect of season between when we sample, and then there's a very strong interaction between the two. So in terms of fish diversity, we see the highest numbers at site 5 and at our Crisp Road sites, and we've seen a couple more at the same site since this was created. And then note that, again, oysters and shelled organisms found at both locations, we tend to see that there's sort of some ecosystem service of those kind of shelled organisms in that environment, along with just general habitat characteristics that overlap with them. And this is just median abundance of fish and invertebrates. You can see there's some sites where we see relatively few. There's some sites where we see, quite a wide range. And, there's our most common species up there. And then if we look at totals in terms of like by species, you just see this overwhelming abundance of Fundulus heteroclitus here. But note, there is one weird exception where at our Crisp Road Salt Marsh reference site, we did catch a large abundance of Pelemon, which are grab string, using a seine. And we didn't really see any anything close to that at most of the other sites ever. So that was very interesting. So I wanted to thank Dr. Anderson, who's my academic advisor. I wanted to thank Asit for doing the drone survey and providing us with wonderful photos. I wanted to thank Ty and Drew and Keegan and Renee, who have all helped me put together the data for this for these surveys. And I also wanted to thank our sponsors, Southeast Mitigation, as well as the landowners of the site, as well as the Edisto Land Trust, which is not on here, but they've been assisting us getting access to certain reference sites. All right, with that, oh, sorry, my citations are on here. And with that, I'll take any questions. Yes and no. For a while, there had been water control structures there that were fully functional, that people operated and like opened and closed, and there was like water going in and going out. Basically, while the structures are still in place and it limits what organisms can kind of move in and out of those impoundments just based on size, they did kind of disable those water control structures so that nowadays there's the tide can kind of just come and go freely. It's just on a different schedule because the water that's entering those impoundments has to pass through a pipe that's about this big. And each of the impoundments, there's only one real like primary connection to the actual tidal creek that they're all connected to. So the water has to go from one impoundment to the next impoundment to the next impoundment, and that results in it kind of getting skewed in terms of the tidal cycle.
What are the history of those ponds? Why were they built?
That's what I was going to ask. What was the intent?
These ponds, I think the most likely scenario that our stakeholders talked about is that they were originally meant to be like waterfowl pond at some point. They mentioned like 1940s, 1950s, because there's a lot of like antebellum rice field ponds that are out here, and it's possible some connection there, but they seem to suspect that these ponds are a bit more recent. And they've probably always been saltwater to some extent, just because of how close they are to the ocean. So they're thinking they probably were used for like waterfowl hunting or something along those lines.
It was a shrimp aquaculture facility.
And it was going to be one, right? And then it was not.
It was.
Oh, it was at one point?
Christopher, on the heels of Dorothy's presentation, are areas like that, in conjunction with oysters, do they have the ability to filter microplastics or PFAS or anything like that?
I was just talking about this. It's pretty interesting. Oysters are very selective in terms of the microplastics that they take up. They tend to take up larger, at least from the research that I've done, like literary research, they seem to take up a lot of larger particles that have a lot of bacteria on them because they'll actually eat that bacteria that's on those microplastics. And generally what happens is it seems to leave them. Sometimes it accumulates within them, but also they seem to be better at a lot of organisms at expelling it. And that's usually going to go into the sediment. So there's always the potential that that microplastic that goes back into the sediment can enter the water column again. But in terms of stuff that's like actively around the water column, I bet oysters are probably good at removing some of the microplastics from it. But there's also particles that they'll just take up and then reject, just like anything else.
Christopher, you include fish and crustaceans together.
Yeah.
I noticed that, like, for some of those, there were a whole lot, like shrimp specifically. So what other vertebrates did you look at, or what other crustaceans did you look at, or did you see?
In terms of crustaceans, we do have blue crab, which have kind of been from their own, generally from their own stuff. I did do crab track surveys, but I've only done like 2 up to this point, and the numbers that we've collected are very, very low. So I didn't think it was worth it putting another graph on here for that. But we do get them in pretty much all of the impoundments to some extent. And then I kind of clustered the crustaceans with the fish because of their association with like human use and stuff like that, where there's like sport fish, bait fish that humans use for multiple purposes. And then there's the crustaceans like the pink shrimp and then the blue crab and the bud crab and some of these other things that have their own uses. Kind of putting them together. I've seen other research papers also do that, where they kind of put crustaceans and fish together. In terms of other invertebrates, the primary thing that you find are mollusks at locations oftentimes where we see oysters. We'll see, I believe surf clams. They're either surf clams or channel shot clams. And then oysters, and then we see ribbed mussels. Ribbed mussels will sometimes show up in locations where oysters aren't because they don't need that hard structure. But yeah, that's why there's not a lot of data here for the oysters in particular yet, just because at these restoration sites, there's not a lot of hard structure for them to colonize. So they're kind of limited.
Thank you.
Thanks, sir.